Good afternoon to everybody. So I'm going to talk about also insects and bacteria, but I'm going to talk about Wolbachia and Wolbachia functional genomics. In the lab at IGC, we are interested in host-microbe interactions, and we have several general questions that we're interested in. How are the interactions specified? How are they controlled? How does the host and the microbe influence each other in terms of physiology, ecology, evolution? And also, since animals and plants normally have a large consortia, how are these consortia regulated? If it's just a sum of one-to-one interactions, or if there are rules in the consortium assembly. So we don't work with humans. We work with fruit fly, Drosophila melanogaster. And we do that for several reasons. One is that the composition of these communities is simpler, although it has some complexity. So it's easier to address some of these questions. And also we can make axenic flies and then reassemble these communities as we wish. And we have the power of the host genetics. And in the lab, we're interested in Wolbachia, interaction with Wolbachia, with virus and with gut microbiota. And I will talk mainly about Wolbachia and a bit about Wolbachia and virus. So there's several reasons also to study Drosophila melanogaster. One is as a model to make fundamental science and fundamental discoveries, but also as a model for other insects. And insects are the most diverse group of animals. So most animal species are insects, and insects can have important impacts in ecology and to humans too. You just heard about a nice insect like bees, and then you also have nasty insects like mosquitoes that can transmit diseases to humans. Now, in insects, it's very common for insects to carry intracellular bacteria that are vertically transmitted from the mother to the progeny. And these are endosymbionts. And there's a huge diversity of these. But sometimes they are facultative, so the insects can live without them. And sometimes these are facultative that are parasitic, or sometimes they are mutualistic. And then some are obligatory, and that is without the endosymbiont, the insects cannot either survive or reproduce. Now, one of the most common, if not the most common, endosymbionts of insects is Wolbachia, which infects up to 2/3 or it's present in up to 2/3 of insect species. So it's probably the most common intracellular bacteria in animals. It's an obligatory intracellular bacteria. You cannot cultivate it. And it's as an endosymbiont, is maternally transmitted. And it was first found by striking phenotypes in the reproductive biology of its host, but can also induce other interesting phenotypes. And one of them is that Wolbachia can induce protection against virus in Drosophila melanogaster, where it was first found, and later on found to be true for other insects. And as you can see here, if we infect flies with the Drosophila C virus, if they don't carry Wolbachia, most of them or all of them die very fast in 10 days. But if they carry Wolbachia, they survive much better this infection. And if you look at the titers of the virus, there's a 10,000-fold difference in viral titers. So there's a strong protection against this infection. And this raises a lot of fundamental and interesting questions about the interaction of Wolbachia with Drosophila melanogaster and its host that we try to address in the lab. But also it has a very immediate applied angle to it, because if you transfer this Wolbachia from Drosophila into Aedes aegypti, which is the main vector of yellow fever, dengue, Zika, chikungunya, then the Aedes aegypti also has a strong resistance against virus, and you can see it here. That there's a huge reduction of the dengue virus titers in the mosquitoes that now carry these Wolbachia. And if the mosquitoes are not infected with the virus, then they cannot transmit it to humans. So there is right now several efforts to use these transinfected mosquitoes in natural populations in mosquitoes, in cities and places where there's endemic dengue or other of these arbovirus, to see if this transmission from human to human through the mosquito vector, if they can be blocked. One of the biggest efforts is carried on by the World Mosquito Program. And the data that is published is promising. There's a lot of unpublished data that is reported to be quite good in terms of efficiency of this strategy. So this has been tried in several pilots in different cities of the world, and just recently WHO has recommended to start a program to deploy these mosquitoes all over the place to see if they can be if they are a good intervention to control dengue. Now, despite all of these excitement with this strategy, we actually know very little about this interaction and how is Wolbachia giving protection against virus and what modulates this protection. And in our lab, we started to work on this very many years ago already. And some of this work was done by a PhD student, Eva Krostek. And we looked at the natural variation of Wolbachia carried by Drosophila melanogaster. So this strain is called wMel. And Eva characterized the protection against virus of some strains that were previously known to have some diversity, some genetic diversity. And as you can see here, all flies carrying any of these Wolbachia variants, they are more protected than flies that don't carry Wolbachia. But there's a difference between these two groups. There's this CS-like group where there's more protection than this wMel-like group. And if we look at the titers of Wolbachia in these flies, you can also see that the Wolbachia that gives more protection also is at higher titers than the Wolbachia that gives less protection. And these results were interesting for several reasons. One is because it shows this correlation between amount of Wolbachia titers and protection. The higher titers, the higher the protection. Also, because we sequence the genomes of these different variants, we also know that they belong to two clades of wMel. So there's also a genetic basis for these differences, and so there's a genetic basis for Wolbachia being able to protect more or less, or be it higher titers or lower titers. And it shows that titers can be important in these interaction and in this protection against virus. So Eva also looked at another Wolbachia that was known in Drosophila melanogaster that was very particular because it was pathogenic. So this Wolbachia was isolated by Benzer lab in 97. And they just found it by chance. They were doing a genetic screen for something else. And they found this Wolbachia that overproliferates in the fly and ends up killing the fly very early. And these are results now by Eva, where the wMel pop, the name of this Wolbachia. So this wMel pop now, it's in the same fly genetic background than this wMel CSB, which is closely related. And as you can see, the flies that carry wMel pop, they die very fast. So there's no virus here, it's just the Wolbachia itself. It's killing it because it's growing exponentially and kills the host. So this is very unnatural because endosymbionts in principle cannot be pathogenic or very pathogenic to the host because it needs to be maternally transmitted. So it cannot be pathogenic to the females that carry it. Because if that happens, it's not transmitted, or there's a fitness disadvantage. But in the lab, we have kept this strain of Wolbachia. Now, Eva sequenced these two strains and these two variants, and indeed they are very closely related. And there was only two SNPs, or two synonymous SNPs between them. which didn't seem very promising as the causative genetic basis for this phenotype of Wolbachia. But there was also an amplification of a region of the genome of wMel pop. So a 23 kb region of the Wolbachia genome was amplified, and we could see this by looking at the coverage of the sequencing data. So it was amplified several fold in wMel pop. So this region contains about 8 genes, or 8 predicted genes to be encoded in this region. And at the time, we put forward the hypothesis that maybe this amplification caused the phenotype of wMel pop, makes them overproliferate. Now, we couldn't, and we still cannot genetically manipulate Wolbachia. So it was difficult to think out how to test this. How could we show that this amplification caused overproliferation? But we also knew that from the literature that gene amplification like this is very common in bacteria. And normally this gene amplification happens in tandem. It's tandem amplification. And because it is in tandem, it's normally unstable. By recombination, you can get increases or decreases in this copy number of duplicated genes. So we thought, okay, maybe there's variation in the copy number of this amplification of this region that we call Octomom. So maybe there's variation, and if there's variation, maybe we can select on that variation, and we can establish lines that carry Wolbachia with high copy number of Octomom, and lines that carry Wolbachia with low Octomom copy number. And if we can establish these lines, maybe we can test the phenotype and know if the copy number of Octomom has an influence on the phenotype. And this was one of the only times that the plan of the project just worked exactly as we wanted. So first, have a look at the stock of flies from wMel pop, and she could see that there was variation. So different flies. So each dot here is a fly, and the copy number of Octomom in the Wolbachia that this fly carries could vary between 2 and here between 2 and 5. Yeah. So evident that pick up the flies that had Wolbachia with the highest copy number of Octomom, and these flies with the lowest copy number, and establish lines with high copy number and low copy number. And at some point, by luck, by fortune, we even isolate one line that reverted from 2 copies to 1 copy. Okay, and then we tested the phenotypes, and so we had this wMel CSB, which is our control line, which only has 1 copy of Octomom region, and we had wMel pop lines, but now they carried Wolbachia either with 1 copy back to wild type, or with 2 copies of Octomom, or with many copies of Octomom. And the growth of this Wolbachia is dependent on the copy number of Octomom. So if wMel pop now reverts to just 1 copy, then it grows exactly as wMel CSB. If it has 2 copies, it grows much more. This is a log scale, so this is an exponential growth. And if it has over 10 copies, it grows even more and kills the flies very fast. So this Octomom copy number also impacted survival, and also impacted protection against virus. So the higher the copy number, the more the Wolbachia grows. The higher the titers, the higher the cost, so the less the flies survive, but the more protection against virus it can give. So later on, a new PhD student in the lab, Luís Teixeira, came into the lab, and we're planning to try to move over from this natural variation and try to do mutants of Wolbachia. Okay, so we thought about trying to do a classical genetic screen in Wolbachia, try to mutagenize Wolbachia, and then screen for phenotypes and find mutants with new phenotypes and identify the mutations and know which genes of Wolbachia are important for what. Now, a problem is, again, we cannot isolate Wolbachia. So what you're seeing here is an embryo of flies. With DNA staining, these circles are host nuclei, and all of these dots are Wolbachia. So if we mutagenize Wolbachia in a fly, that means that each new mutant we generate, it will be a single cell of Wolbachia in the middle of all of this population inside the host. So it will be very difficult to assign a phenotype to this mutation. It can be diluted in the middle of the population of Wolbachia. But there could be one circumstance where we could overcome this, which is if we could hit this Wolbachia population with a mutagen, and one of these new mutants started to overproliferate, then maybe it could take over the population of Wolbachia in the host, and we could detect this overproliferation by quantitative PCR. And we had some good reason to think that was possible, because also in a previous work by Eva, we saw that as a flies age, flies that carry wMel pop, the copy number of Octomom increases with age. Okay, and our interpretation of this result is that over the life of the fly, there may be selection, within host selection, for the Wolbachia that replicate more. That is, for the Wolbachia that has a higher copy number of Octomom. So the experimental design for the screen was this one. So we had flies, females carrying Wolbachia, and we gave them a mutagen EMS. We let them lay eggs for a few days, and then we pick up the next generation of females. So these females, they may carry Wolbachia that is mutagenized, and it was mutagenized in the mother and transmitted to them. Then we let them lay eggs for a few days, collect them. Elvis did this, and do quantitative PCR for each individual fly to see if any of the fly now has a higher titer of Wolbachia. If we could identify such a fly, then we had the vial where the eggs were laid, where the progeny was developing, and follow up this progeny and try to isolate this Wolbachia. And the screen actually worked. And out of approximately 1,000 lines that were tested, Elvis was able to detect 2 lines of Wolbachia that overproliferate. And you can see here the titers compared with control. Now, the protocol may also lead to mutations in the host, but we can replace all the chromosomes of the host by genetics. And what you see here is already the titers of Wolbachia in flies that have exactly the same genetic background. So this is due to Wolbachia differences and genetic differences in Wolbachia that are responsible for this overgrowth. So next we sequence this Wolbachia to understand what would be the causal mutation giving this overproliferation. And one of the lines only had one difference between the ancestral control wMel CSB and this new line, and that was amplification of a genomic region. And this is exactly the same genomic region. Okay, so we replicated or better, we confirmed the result of the wMel pop by, instead of going from a pathogenic wMel pop back to a non-pathogenic one by reducing Octomom copy number, we started with a CSB normal Wolbachia and show that it becomes pathogenic when this Octomom copy number increases. And we call this line wMel pop 2. Now the other line had only one difference with the ancestral line, and this difference is a deletion of exactly the same region. Okay, so either amplification of this region or deletion of this Octomom region leads to overproliferation. And although it's similar that both of them give rise to overproliferation, their phenotypes is not exactly the same. And Elvis did a very detailed characterization of the phenotypes of these Wolbachia, and I'll just give you a few of these data. So we could see that this wMel pop 2 overproliferates and grows exponentially during adult life. And actually, it's not shown here, but there's exactly the same phenotype as wMel pop. And wMel octoles also has higher titers than control, and it grows a bit more than wMel CSB. Now, what's also interesting is that this is the data of growth during adult stage. Can be very different from between these variants of Wolbachia. But we also look at the growth of these variants during development of the larvae. So we look at these titers throughout the development stages of the Drosophila monogaster, from the egg to pupae and then to adult. And what we could see was two things. First, that there is a very fast growth of Wolbachia from the egg until the pupae, and then it seems to slow down or stop during pupal stages until they are adults. Okay, and the second thing we saw is that this is true for all of these Wolbachia, for these new mutants too, and the growth rate is very similar between them. Yeah, so this CSB that doesn't grow much in the adult flies, it's actually capable of very fast exponential growth, while the flies develop and as fast or very close to the same rate as wMel octoles and wMel pop2. So these differential phenotypes between these mutants, it's also dependent on the stage of development of the host. And if we calculate the doubling time of these different Wolbachia, you can see that they're all very similar during the larval stages, and much faster this growth than during adult stages. LBs also measure growth during adult stages at different temperatures, and there's interaction between the temperature and the different strains. In general, Wolbachia grows faster at the higher temperature, but not all the variants respond the same way. And wMel pop and wMel pop respond very strongly to temperature, and they can grow very fast at high temperature. Now, this is all about growth. If we look at the effect on the survival of the flies, we see that all of these new variants, they impact on the survival of the flies, and they die faster. But also there's interaction between these and temperature. Their impact on the host differs with the temperature. Now, with all of this data, we could also look at the relationship between pathogenicity of Wolbachia, protection against virus, titers, and growth rate. And we could see that this pathogenicity of these mutants correlates very well with the doubling time at the different temperatures. Okay, so the more the Wolbachia grow, the faster the Wolbachia grow in adults, the stronger the impact on the survival of the flies, and there's a very good correlation with that. If we look at the protection against virus, you could see that all of these new mutants that overproliferate, they protect more than the wMel CSB, but it didn't correlate with the growth rate. Yeah, so those correlations are not significant and are quite weak, but it correlates very well with the titers of Wolbachia at the point of infection. So this is interesting because it's showing that this benefit, which is antiviral protection, and the cost that is shorter survival, they correlate with similar traits, which is titers and growth rate, but not exactly with the same traits. So maybe it's even possible in terms of application to try to find Wolbachia that give more protection, but don't have a very strong cost to the host. Okay, and this is also interesting because in mosquitoes, in fact, there are two Wolbachia that were transinfected into the mosquitoes. There was wMel, which is the one that is being used nowadays in the field, and wMel pop, which originated from wMel pop, but it also overproliferates strongly in the mosquito, has a high cost to the mosquito, so it doesn't spread into the population when you try to release these mosquitoes with wMel pop. And when this strain was sequenced, it was actually seen that it had a deletion on the Octomom region. Okay, but because it has a deletion on the Octomom region and it overproliferated, then it was also put forward the hypothesis that the Octomom region had nothing to do with overproliferation. But what we're seeing with our results is actually it does. It's just that either amplification or deletion, they both lead to overproliferation. Okay, so in this region, there's a series of predicted genes, and we'll be very interested in trying to understand which of these genes are important for these phenotypes, and if which genes are important in the gain-of-function phenotype and which genes are important in the loss-of-function phenotypes. They could be the same or different genes. And just to conclude, what can we take out of this as a message? That we can actually do genetic screens in Wolbachia, that this amplification or deletion of Octomom region leads to overgrowth, that this growth is due to interaction between different factors, the Octomom copy number, the temperature, and the host life stage, that the cost and antiviral protection correlate to slightly different things, and we could play with this. We can show that this region is not common in all the Wolbachia strains in different insects. It's actually not present in most, but it's present on wMel, the Wolbachia of Drosophila monogaster. And there it seems to be an addictive genomic island, because now you cannot remove it, because if you remove it, they overproliferate. That has a cost to the host, and therefore a fitness cost to Wolbachia. And overall, we can do some functional genomics and functional genetics. And these wMel-pop, wMel-pop2, and Octoles are the first and the unique strains where we really know the mutation and the phenotype associated with these mutations. So I just want to finish by thanking everybody that did this work, the group, when we could get together before COVID. Eva that did the first part of the work I talked about today, and Elvis that did this second part of the screen. Funding agencies, all of you for being here. And I just want to finish by again just stating that there will be a next seminar of this series on Thursday of 20th of May with Heinrich Schulenburg and Karina Xavier, that you can register. There will be a register page. If you have doubts, you can write to this email. And also, if you want to speak with me or Philippe today or tomorrow, we have some time for that, and you can write to this email. Okay, thank you.
Thank you, Luís, for the very nice presentation. So I have a question. So you mentioned that with this region, so if you have more Wolbachia because of this Octomom region, the flies survive, so they live for less time. Do you know if there is any effect on the amount of progeny that they have, if they have less, if they put less eggs, or if they reproduce less when they have this region or not?
Yeah, we look at this and we don't see obvious phenotype on it. I mean, they live less, so they will not lay eggs from the point they die on. Yeah, but during their lifetime, they seem to put a similar amount of eggs. Yeah.
Okay, thank you. Does anyone else has another question?
Thomas Bosch.
Hi, Thomas.
Luís, hello. Sorry, or if Philippe, I was late. Luís, question maybe very naive from an outsider, of course, but you talk constantly, and all the community talks about Wolbachia strains. What is that? Is there a systematic behind?
Yeah,
how many strains are there? Do we know what we talk about? Wolbachia is obviously a single species in this genus, but I mean, strains are getting more and more important. So what about Wolbachia strains?
Well, I think it's difficult to know what is a species in bacteria. I suppose it's even more difficult to know what is a strain. Yeah. So the nomenclature that is normally used for strain is a Wolbachia from a different host. Okay. And sometimes you can have inside the same host different Wolbachia that are distinct in terms of genome, that we call them different strains too, or that belong to different parts of a phylogenetic tree, if you do a phylogenetic tree of Wolbachia. So Wolbachia is not very well, in terms of taxonomy, is a bit of a mess. So the species is Wolbachia pipientis, but nobody uses the species name nowadays. We just call it Wolbachia. And it can be quite diverse in many different hosts. and then the strain is applied to these one Wolbachia type from one species. We call it a strain. Yeah, and then on top of that, I and maybe others, I call a variant a different isolate. Not an isolate because we don't isolate them, but a Wolbachia with a different genome that is being carried by a stock of flies. Okay, I call that a variant.
The question I'm asking that is, and I'm making very short, but in the conventional microbiota, we tend to think in terms of function, and so one could group strains according to joint function, common function. People are talking about guilds in the meanwhile, and species is not really important. Species is not important. Important is the function. I got the message.
It's not clear in the sense that you can always break it down. There is, if you look at all Wolbachia together, there is a horizontal gene transfer between them. Okay, but within a host, there is not. There's only vertical. Shorter time span. There's only vertical transmission. Okay, so there's not much horizontal transmission happening constantly. So it's a bit different. Yeah.
Okay. Thank you, Thomas. So we also have a question from André Mateus. Are there closely related species to Wolbachia that are not obligate intracellular and can be genetically manipulated so that you could express Octomom?
Yeah, so the Wolbachia belongs to the Rickettsia, and most of them, if not all, they are obligatory intracellular bacteria. I think there may be a couple of them that can be cultured. Yeah, I didn't think about putting Octomom there. I think we could try to see what's the effect of this Octomom. I mean, it's quite a big region on the bacteria itself, but on interaction with the host, it will be more difficult because I suppose that other bacteria would not be able to colonize the fly. Yeah.
Okay, so one more question from Carlos Vogt. Thank you, Luís, for the interesting talk. Is it possible to do in vitro fertilization with Drosophila oocytes that carry Wolbachia?
Okay, so the short answer is no. We cannot do in vitro fertilization of flies nowadays. That is not possible. And one of the main reasons it's difficult to isolate the sperm cells because they have huge tails and they entangle. If you try to isolate sperm, the tails of the sperm cells just all entangle. So you cannot do in vitro fertilization at this point of Drosophila.
Okay, thank you. So does anyone else has another question? Okay, so I think there are no more questions. So I would like to thank both speakers for the very nice talk and for all the people that attended these seminars. And don't forget that 20th of May we'll have the second round of seminars. So, and that's it. Thank you all and hope to see you again soon.